This recorded session is dedicated to our pre-lab discussion for our module on osmosis and cellular transport. Um, we're going to incorporate our discussion so far with regards to the chemical composition of the cell, particularly the biochemistry of the cell membrane. So at the end of the module, there are three learning objectives that we aim to achieve. Number one, you must be able to recognize or understand osmosis in the context of um, diffusion of particles, diffusion of molecules um, through a semi-permeable membrane. Second, you must be able to understand osmosis by analyzing the effects of different saline solutions to the structure of red blood cells. So this is our laboratory proper. If we are to have our on-site classes tomorrow, then you'll be able to perform this. If not, an alternative activity will be carried out in virtual setting. And lastly, you must be able to explain the significance of osmosis in living dynamic systems. So a few concepts to discuss. I have a few concepts that I wanted to discuss first as prelude to our laboratory proper. So here you can see a usual scenario in our in the Philippine commute system or Philippine commute environment, that on a daily basis, okay, people use public commute like the train to go from one place to another, you know. And in the context of animals, you know, at the cellular level, materials are transported, molecules are transported. Okay, let's review the biochemistry of the plasma membrane or the cell membrane. As a reminder, or as a recall, we have now been introduced to the idea that the plasma membrane serves as the boundary between the ambient, the external environment, and the cytoplasm, the inside of the cell. Uh, in terms of the biomolecules that make up the plasma membrane, largely the plasma membrane is composed of phospholipid molecules which are arranged in a bilayer. And you will remember that aside from these phospholipids, there are proteins that are embedded in the plasma membrane. No? The you can we can again, just a review, okay? So the bilayer is consisting of these two layers. So this first layer here facing the ambient, and this another layer here that faces the interior of the cell, okay? The polar portions of phospholipids, the heads, okay, they are oriented in two directions. The outer layer faces the external environment, while the inner layer or the interior layer have phospholipid molecules with their heads oriented towards the cytoplasm. Okay, and as a review, this is the chemical structure of a phospholipid. Okay, you can see here the phosphate group attached to fatty acids. Okay, attached to the long chains of hydrocarbons, right? And there is, of course, a backbone that serves as the attachment site for the fatty acid chains, the long chain fatty acids, and the phosphate group. Okay. Um, I've already introduced the concept of fluid mosaic. In the past, there are a number of theories that are used or that are conceived to explain the nature of the plasma membrane, to explain the biochemistry of the plasma membrane. And the long-standing theory or model is the fluid mosaic model that describes the plasma membrane as a structure with a dynamic pattern, a mosaic pattern, because the proteins that are embedded on it and the phospholipids, they are not stationary, and rather they are moving. They are not static. They're dynamic molecules that do not stay at a particular point or a particular location for the entire duration of the life of the cell. No? So, the proteins that are embedded in the cell membrane may be of different types. I've mentioned or discussed these types of proteins in our lecture class. And to enumerate or reiterate them, we have channel proteins, transport proteins, we have cell recognition proteins, proteins that serve as receptor sites for certain ligands or molecules, proteins that catalyze reactions, enzymatic proteins, and proteins that connect one plasma membrane or one cell with another, called junction proteins. So we're discussing these proteins because um, we're the primary idea or the primary function that we want to look at in the lab proper, is how molecules are transported in the cell or in and out of the cell or through the plasma membrane. Okay. Now, how do cells transport materials? Okay. So there are two basic types of molecule or chemical transport. So there's passive transport and active transport. Passive transport is the type of molecular transfer that does not require energy, and because of the fact that it follows the concentration gradient, that the molecule moves from a place of high concentration to a place where its concentration is lower. On the other hand, active transport is the opposite. So it goes against the concentration gradient. The molecule is transferred from a place or from a location of lower concentration to a place with higher concentration. Okay? So I want you to think of these places or locations that I'm talking about as the interior and the exterior of the cell. Okay? Right, now let's compare different mechanisms of cellular transport that are generally known. Okay, so we have three mechanisms. We have simple diffusion, facilitated diffusion, and active transport. So in a way, you can say that simple diffusion and facilitated diffusion are two types of passive transport mechanisms. Okay, while active transport definitely requires energy. So what substances are transported via these mechanisms? So in simple diffusion, we have small polar molecules that can easily be transported, like water and glycerol, gaseous molecules, small non-polar gaseous molecules like oxygen and carbon dioxide, and non-polar molecules, which may be large or most of the time large, like oils and steroids. No? Facilitated diffusion, on the other hand, carries out the, the transfer, the movement of larger polar molecules like the carbohydrate glucose, no? And also charged substances such as charged molecules such as ions. Okay, and similar with facilitated diffusion, active transport also caters to the movement of larger polar molecules and those with active charges. Okay? In relation to the concentration or electrochemical gradient, simple diffusion and facilitated diffusion is always following that. down, so meaning from higher to lower, while active transport is the opposite. No? There's no requirement of energy, and with there is intrinsic directionality for the passive transport mechanisms, while for active transport, energy is required, and there is somehow an intended direction where the molecules are transferred, are expected to be transferred. No? In terms of kinetic properties, membrane proteins are required in active transport and facilitated diffusion, while in simple diffusion, the molecules just pass through the phospholipid layer without any assistance from any other kind of molecule or protein, to be particular, no? And then in terms of the saturation kinetics, simple diffusion does not really follow that, while facilitated diffusion and active transport actually follows or has saturation, observes saturation dynamics that at a certain instance these processes can stop because of the achieved concentrations or the relative concentrations of the molecules that are being transported from one location to another. In terms of competitive inhibition, in relation to saturation dynamics, also facilitated diffusion and active transport follows competitive or displays competitive inhibition. Okay. Now let's take a look. We're using more illustrative examples. So simple diffusion, you can appreciate that from a location of high concentration of a certain molecule, okay, the net movement is towards the area where its concentration is low. So from high to low concentration. Okay. And again, there's no need for any assistive or supporting molecule to transfer these compounds or these molecules. Right. Then, so I mentioned this already, net movement is always from following the concentration gradient. And there is no energy, ATP, that is involved. Okay? The idea, of course, for simple diffusion, no, an important concept I'd like you to remember is the concept of equilibrium. Okay, molecules will move along the concentration gradient as long as the concentration of these molecules are not equal in both sides. No? So once that the concentration of the transported molecules is equivalent in both sides, okay, of between in both sides, one in the exterior or the internal environment of the cell, that's the time that diffusion will come into a halt. No, it will stop. Yeah. Now, moving on to another kind of passive transport that does not require energy, we have facilitated diffusion. But unlike passive transport or simple diffusion, facilitated diffusion needs to or employs assistive molecules, transport proteins embedded within the membrane. So transport proteins that are integral proteins. Okay, there are several types of facilitated diffusion that can be carried out by carrier proteins. Those that can transfer molecules from the outer environment to the inner environment, or vice versa. We call them uniporters. Okay. On the other hand, we have coupled transporters. We have symports and antiports. So these proteins are able to transport molecules, two types of molecules simultaneously. Okay, for symport, see these proteins are able to transfer molecules from one direction, two molecules from one location to another. While for antiporters, the direction or the transfer or transport of the molecules is in opposite direction. So one goes inside the cell and another goes out. Okay? Right. So just to have a nice visualization. So here you can see an integral protein embedded in the cell membrane. And the transfer of the molecules is facilitated by these integral proteins, by these membrane proteins. No? So high concentration of a lipid insoluble solute. So let's say, for example, we're trying to transfer sugars or amino acids, no? From an area of high concentration to an area of low concentration. Since these molecules are lipid insoluble, and by now you already appreciate that a big chunk of the plasma membrane is made up of lipids, made up of fatty acid, of hydrocarbon chains. Okay, so the simple diffusion of these molecules will be unlikely. Okay, they have to be carried by another molecule. They have to be carried by membrane proteins. Okay, so these membrane proteins embedded in the cellular membrane, as I already discussed, have several variations or types. Okay, so here you can see that after a molecule, a lipid insoluble solute is transported. Okay, there's somehow a change in the position or the conformation of the membrane protein. Okay, in order to release that solute that was captured from the external environment, in this example, to the cytoplasm or the internal environment of the cell. Okay. Now, let's take a look at osmosis. Okay. So, for technically, we have to define it. We have to reiterate. We have to mention the technical definition of osmosis. Osmosis is the net movement of water molecules. So a while ago, what you have seen so far, are solutes that are transferred, no, from the ambient to the cytoplasm or vice versa. Now, for the process of osmosis, or in the process of osmosis, what is moving are water molecules. Okay, what moves are water molecules. And it's important to make mention of the fact of the qualifier across a semi-permeable membrane. So there is a qualifier, right? So the molecules of water moving across or through a semi-permeable membrane. And as a passive transport mechanism, okay, the the movement of water, or in relation to passive transport, the movement of water is in reference or dictated by the concentration of solutes. So please remember this. Okay. So during for osmosis, or in osmosis, no, yung theory, right? So water moves from an area of um, lower solute concentration or lower solute activity to an area of higher solute concentration. So the idea is, or essentially think of water, the tendency of water, which is, or the property of water, which is to always dilute material, always try to distribute the concentration to make sure that the concentration is equal. No? So water does that. It has the natural propensity to be attracted to a location, okay, where there is a higher solute potential that needs to be diluted. Okay, so it always goes to a location where dilution is needed. Okay, and again, the context is across the semi-permeable membrane. Okay. Here in this illustration, so you can see that water molecules are represented by the blue and yellow circles that serve as representation of one water molecule. No, so remember that water is actually partially charged. No, so in our comparative table a while ago, I mentioned that, or we've discussed that molecules with net charges, positive or negative, can find it hard, no, to traverse the cellular membrane. So for the case of water, even though it's just possessing partial charge, okay, it might still have some challenges diffusing simply across the plasma membrane. But as the universal solvent in our planet, okay, water still diffuses across cell membranes, but they diffuse very slowly. So remember that, okay? Right. So another concept that we relate to osmosis is tonicity. So tonicity refers to the ability of a solution to change the shape or tone of cells by altering the internal water volume of the cell. Okay. I'm not sure if you had introductory biology lab, but if you did not, then probably definitely this is the first time that you're hearing about it. No? So in both animal and plant cells, okay, there are different solutions that can be looked at or experimented on, and we can see how these solutions affect the tonicity or affect the structural integrity of plant and animal cells. No? So I'll be introducing these three solutions. We have hypertonic, isotonic, and hypotonic solutions. So, hypotonic solutions are solutions that are more dilute than the internal environment of cells. So, when cells are placed in a hypotonic solution, they enlarge because water will rush inside them. Okay, so remember, during osmosis, the tendency of water is to go to a location where there is higher solute concentration. So if the ambient is hypotonic, then water will go inside the cell and will try to equilibrate, okay, the concentration of the solutes of the internal environment and the ambient. Okay. The second type of solution is isotonic, which has the same concentration of non-penetrating solutes as those that are found in normal living cells. So isotonic solutions, the net movement of water is at zero. So whatever amount of water molecule goes inside the cell will go out as well. So definitely there is no change in the integrity, the appearance of cells that are subjected to isotonic solutions. So the word iso meaning equal. Then we have hypertonic solutions, which have higher concentrations of non-penetrating solutes than those that can be seen inside the cell. So when cells are immersed in hypertonic solutions, the tendency of water is to go out of the cell, and the cell will lose size or will lose volume, will have decreased volume, and will shrink. Okay? So for animal cells, it's very much demonstrable, but for the case of plant cells, it's quite hard or challenging to visualize them when you're observing plant cells, because remember that plant cells have cell wall that are very rigid. So the cell wall ensures the structural integrity or volume of plant cells, even though they are subjected to either hypotonic or hypertonic solutions. Okay? So just as a review, or I mean to emphasize for plant cells, you can see that the cell wall, of course, will not be affected, but the cells surrounded by the cell wall will still manifest the expected changes in its size depending on the tonicity of the ambient solution. Okay? For your laboratory proper, if we are going to have an on-site class tomorrow, so you'll be, I'm hoping this time we read through the instructions and we will be able to follow the instructions as faithfully as possible. So, uh, during our last meeting, you extracted some blood from your classmates and then observed the blood cells under the microscope. So this time, those blood cells, we will be subjecting them into different types of sodium chloride solution, different concentrations of NaCl solutions. And your goal is to be able to observe and take note of the differences in terms of the cell morphology, the appearance, size of these RBCs that are subjected into different the different salt solution concentrations. Okay. If you have questions, we will be talking. We can discuss them during our laboratory proper. Okay.